Let's begin with hubs. So we have protein-protein interaction hubs. What's your association with that? If I throw that term at you, again, I totally understand that you don't know what it is, but what is your association with that? Any association with that?
I'm thinking about a network hub.
Yeah.
So it's like one controller in the network which routes traffic to all associated connections.
Yes, and I guess that's back to this idea of a network, that a network has a structure. And one aspect of a structure is that some is sort of, you say, is a root. It's a sort of a thing, a hub that plays a special role. Now, let's just look at the world of biology. So this is the way we see that often in technical networks, played out in technical networks. What do you believe in biology? Well, proteins have a similar number of interactions, and sort of a network theory, number of interactions that a particular protein has, typically you call the degree. And then you talk about hubs, proteins that sort of have high degree or something like that. So if we looked at the plot of degree versus number of interactions or distribution here, what essentially I show you is three different extreme models. And you could have had all kinds of things in between. A straight line, so essentially the idea that all sort of probability of, there's a certain given probability, or I could have had this one too, I forgot about that. So the solution could have been that all proteins have a certain degree. So that would be a peak somewhere here. Well, let's just call it a much sharper Gaussian. So one idea here in blue is everything has sort of the same, every degree is represented by a similar number of proteins. And in the normal distribution, again, where you focus it, this is not really the issue, but the idea is there is some particular degree that many proteins have, some have fewer, some have more. And then there's this sort of green line that essentially says some proteins have a lot of, some proteins don't have many interactions and others have a lot. So in some sense the green line is the kind of thing that you would expect when you talk about hubs. So what's your expectation for proteins? Do you have any expectation?
Green one.
The green one. So something that many proteins have very few interaction partners and some have a lot, which essentially is a hub. Why do you say that?
Because proteins are interact with each other, but for a specific protein, they only interact with a specific protein partner.
Well, that could be compatible with the idea that essentially every protein has one interaction. So that could be compatible with the idea that, you know, we have a Gaussian that is very close to here, right?
But some protein tend to have many partners.
Okay. And so out of that comes something like that. So that's a good point. So an observation that if I know there's a substantial population here, and I know that well, actually then we could have a U.
Yeah. But that would entail that we have a lot of proteins that have many interactions.
Yeah. Well, actually, I sort of put that into your mouth. It's true. You said some proteins have a lot. So that is exactly the counter argument against the. If that really is your image, then that is the counter argument against the U. Now, any idea? Is there any system in the world that you know of, other than man-made systems, for which you somehow know a distribution of this type? Let's not only think about network, let's just look at it in a sort of a generalized way. Let's ask the question differently. If you plotted the distribution of settlement size, so settlement is essentially a village or whatever you call that, right? So if we plotted on this planet the size or the number of inhabitants in any sort of distinguishable unit, Let's call this unit a settlement. And settlements in this language, they go from megapole cities, whatever the top at the moment is. What is the biggest? Is Beijing's the biggest at the moment? Tokyo no longer is the biggest. Tokyo, I believe, was surpassed. I believe even Shanghai is bigger than Tokyo now at the moment. So for most of my lifetime, Tokyo was the winner in this. But anyway, so if we plot this, how do you believe settlements look? Any idea?
Gaussian.
They would look like Gaussian. And in fact, the remarkable reality is they don't.
Yes.
The remarkable reality is, this is when you plot, and it goes back to Zipf's law for cities is here from this publication. Zipf essentially is a power law distribution, the logarithm of which is a straight line. Or the log of, it depends on how you plot it, the direction that you plot it. But ultimately, so this is a log. So ultimately we have the situation that you have a growth in competition. So if you programmed in the computer something where you put essentially that you have a competition, so competition for size. So if you sort of randomly distribute dots and you have a competition and you have some attractive force, then what you actually create is this distribution. And this is, so you don't need a lot of assumptions in your toy computer program in which you can actually create that to create the Zipf law. And that is why most likely the Zipf laws are observed in many situations, such as the settlements, such as bridge sizes, such as word lengths, word numbers in sentences, and all kinds of things like that. And that in fact was one of the Mr. Zipf observed it for words. So this is the Zipf law, that's the distribution, the raw distribution. So this is essentially 1 over x. And this is roughly the way it looks. So now that means that in fact for our protein interaction, what we expect is something like this yellow one. Now the question is, if you picked, so we know that when protein interactions are measured, we are not having a complete measurement of human. Okay, we know a lot of interacting networks, proteins, so we can construct from these proteins networks, but we know this network is not complete. Now let's assume that whatever we find is sort of a random bias. So I pick randomly from the entire network. So I know the entire network will most likely be Zipf-like. So the question now becomes, if I picked randomly from a Zipf, what do you get? Any intuition for that? Let's turn that around. You all are much more familiar with the Gaussian distribution. If you pick randomly from a Gaussian, what do you get?
The mean? Somewhere in the mean?
No, sorry. I did not formulate my question right. So you have a Gaussian distribution of points, and now you randomly remove 10% of the points. What is the remaining distribution of the 90% of the points looking like? It's a Gaussian. If you do it again, this is a Gaussian, right? At some point, you're going to have so few counts that you cannot recognize it. You cannot see it as a Gaussian anymore, but the subset of a Gaussian, a random subset. So if you picked from a Gaussian some extreme ends, immediately you would change it, right? But a random subset of a Gaussian, and ultimately, your answer that ultimately you will get at the mean, and on average you will actually get at the mean. That's also a correct answer. In the same way, actually the subset of a Zipf is a Zipf. Okay? Now, ultimately now that means that if from this set of interactions that we observe, we have a Zipf, then all we know, it will not tell us anything about the missing ones, but it will tell us something, namely that the set of interactions that we have essentially is a random subset, right? That's all it tells us. And in fact, that's what we see. Now let's go back into a different story. I talked about on Thursday a method that predicts binding sites. takes a sequence and it predicts binding sites. It does not predict interactions at all. It does not actually predict numbers here. How could we relate that prediction of binding sites to numbers of interactions? Any idea? How could we try to see whether this prediction of molecular binding sites, molecular, I call it molecular because essentially the binding site is a residue that binds another protein, while the protein binding another protein is sort of more macromolecular. It's between two molecules, right? And it essentially determines is a feature, the degree is a feature of the entire network. So how could we get from this little side of binding site residues to the macromolecular feature of network feature? Any idea?
I mean, one really naive approach would be to distinguish the different binding sites. So, I mean, 3 residues which are classified as binding sites, 3 consecutive residues are called one binding site,
yes.
for example.
It's, by the way, it's by no means naive. In fact, let me call it differently. It's even highly non-trivial. If that were my protein, and if I predicted a binding site here, and a binding site here, and a binding site here, and a binding site here, if you saw that this way, in this pseudo 3-dimensional view, you would suspect that all of these 4 points here, let's just say that each one of these comes with a bunch of predictions, right? That all of these predictions here group into one common binding site. But if we start from sequence, for most proteins we don't have the 3-dimensional structure. So going from the sequence, predicting on my sequence level that these 3 things bind, and these 3 things bind too, does not mean that I know they're closest to each other. And it could as well be that in fact they fall on different sides of the proteins and in fact are different binding sites. So that we will not, so what you call naive is very, very non-easy to do. Highly non-trivial.
Yeah.
But you're on the right track, right? In the simplest way, this is typically what you do in science. So what I can predict is these numbers here. Let's just see whether it correlates, right? And that in fact is work from Abner Schlesinger and Jana Jofran. So in principle, if I saw a correlation in an even more naive way than what you say, what we would expect is somehow we predict more binding sites, we expect more interaction partners. Okay? And there is some, well, do you immediately see, so that of course is one issue here. I cannot resolve. Maybe there is an interaction site that is common in 3D where things come together in 3D on the level of sequence. I don't see that. But somehow I would, on a statistical level of all proteins, I would assume that I can somehow capture that, because I would somehow assume that if the number of binding residues is roughly the same that I predict and observe, although sometimes I make a mistake, then I would say that most binding sites have sort of 20 residues in them. And if I only predict 3, then most likely that's part of a different, so somehow this correlation would still somehow hold, right? But what is an immediate problem with that idea? So again, the idea being, I want to relate what I can predict, what I can measure is this micro level here, number of binding sites, I can predict them. I make mistakes, but at least I can sort of predict the number, and I want to relate that to how many proteins do I bind. And my argument is, if I have more binding residues, then I have more binding partners. So if I have more than, say, typically a binding site has 20, if I have 40, then most likely I have 2 binding sites, so I have 2 binding partners. So on average, there are many things wrong, and ultimately this 3D to 1D story is one thing where it's wrong. But what is another more systematic problem here? And I've sort of played con man here. I sort of, in some sense, deliberately confused you. But that's the idea, right? I have 40 residues, say binding site is 20, I have 40, I believe it's 2 binding sites. So it's binding 2 things, and that's where the con artist comes in. I'm deliberately, with that statement, I'm deliberately misleading you. Why? Yeah?
But is it possible that a partner bind more than one
Exactly. That's exactly the point that I'm misleading you with. It could be that, in fact, this particular one binding site that I see there binds more than one protein.
So this is not what...
So it could be that more binding sites are more binding partners, and it could be that one binding site has many. And that, in fact, is this distinction that goes back to, oh, the quote is not on here. Oh, here it is. A paper from Marc Vidal, where Marc Vidal introduced this sort of two types of hubs, the date hub and the party hub. The date hub is one where you exactly have what you said, Sylvia. You have one binding point that binds many partners. It's like you go to a date and you talk to many people, one after the other, right? While the party hub is the idea, you're standing somewhere at a party and you're talking left and right with different people, so you have many binding sites. Okay? And then we know from the protein world, clearly these two things exist. So the party hub is the typical, let's give it the name that people use, a transcription factor. A transcription factor has to do with reading the genetic information, and while this is done, the transcription factor has to be bound to other things, and that's essentially the way to control. So this way the same transcription factor can do many things by having different control mechanisms, meaning by having different bindings at the same time. So that's why these kind of things are typical party hubs. While date hubs are molecules, for instance, there's a protein that is called ubiquitin that takes proteins that are no longer fit to do their job out of the cell and sort of brings them to digestion. And this one recognizes essentially the same thing in many different proteins, something that is slightly misfolded. That is one way of looking at it. This is an oversimplification. But ultimately, ubiquitin is a protein that has, like a date hub, with one binding site recognizes many different proteins. Exactly what you said, Sylvia. So then what we would expect here is that for the party hubs, so many binding site, we would somehow see a correlation, and for date hubs we would not. So in fact that's exactly what we see. So this is the 3D view, here's the 1D view. So you see that purple and the gray are completely on the same line here. You cannot even, here you see the gray line coming underneath a little bit, but ultimately it's the same line. So that's what you sort of recognize here in 3D, in the pseudo 3D way of looking at it. And the other one goes up. Okay, it's not a linear correlation, it's not a clear signal, there are all kinds of things that go wrong, but you see a signal. And it's actually remarkable in its clarity. So the other part is that the ones where the date hubs go up, we see disorder briefly in the last semester. No, we actually didn't talk about disorder. But so essentially this is a way, did we?
Yeah.
We did talk about disorder. So disorder is a mechanism by which a particular protein can sort of adopt slightly different conformations, and that is a way by which you assume that you get many binding sites, one binding site with many proteins by slightly changing that binding site, and you see that where you see disorder, in fact the date hub is overrepresented. So slight disorder, or a particular type of disorder, it looks like long loops that doesn't have regular secondary structure, neither helices nor beta strands. That's exactly the kind of thing, if you look for that, you see that is overrepresented in the group of all the date hubs. And then there are particular examples where we sort of can see that, where the interaction sites are either, in this particular case here, so wobbly, unstructured, or where we in fact have a complex of a large protein that has many interaction sites. And we see both from the sequence. We can predict both from the sequence.